What's up, biology students? Well, you know who this is by now. In today's video, we're going to talk about the structure and function of chromosomes. In our last unit, we learned a ton about DNA and what this important molecule does inside of our cells. Well, chromosomes are the structures that hold our DNA and our genes, and our genetic traits are carried on chromosomes from one generation of cells to the next. Our DNA is organized into chromosomes just prior to cell division, and this helps to ensure that each new cell produced by cell division gets the proper amount of genetic material. That's because it's a lot easier to evenly split up the cell's DNA into two equal portions if all of those 3.5 billion base pairs that make up our DNA are organized into easily movable, compact structures. In eukaryotes, which includes organisms like us, chromosomes are found in the nucleus of the cell. Prokaryotes have chromosomes as well, but they are much simpler in structure, and they are found freely floating in the cytoplasm of the cell. In eukaryotes, in the nucleus of the cell, DNA is wrapped around these proteins called histone proteins, and the resulting structure is called a nucleosome. These nucleosomes, which resemble beads on a string here in our picture, are tightly coiled into a filament, and this filament is then coiled into an even larger filament, which is then tightly coiled into a chromosome. All this coiling and recoiling means that you can pack a lot of DNA into a very compact structure, which of course makes it easier to make sure that this structure ends up in the right place during cell division. This X-shaped chromosome that we see in this image is a duplicated chromosome, which means that the DNA inside the cell's nucleus has already been copied by way of DNA replication, a process we learned about in our last unit on genetics. Each arm of this duplicated chromosome is identical to the other, so each arm is called a sister chromatid. These sister chromatids are held together in the middle at the centromere. During cell division, these sister chromatids will be pulled apart and separated so that each new cell produced during cell division will inherit one of these two sister chromatids. And because these two sister chromatids each have the same genes, that means that each new cell will have the same set of genes as the other. This diagram depicts an unreplicated chromosome, meaning that the DNA in this chromosome has not yet been copied by way of DNA replication. This diagram also depicts another important region found on each chromosome called a telomere. Each end of this chromosome is capped with a telomere, which contains a unique sequence of bases that are repeated several times. These telomere caps do not actively code for proteins, but they do protect the active DNA contained in the chromosome from being broken down and degraded by enzymes in the cell. Over time, these telomeres degrade and become shorter and shorter, and this degradation is associated with the aging process. As you can see in this graph, our telomeres at birth are about 5 times longer than they are at age 65. This is why we get old, scientists think, and why our bodies start to break down later in life. Because as our telomeres wear away to nothing and we lose the protection they provide, it becomes more likely that errors will occur during DNA replication and that our active protein-coding DNA will start to degrade as well. So our telomeres are important because they protect our genes and the ability of the cell to manufacture the proteins that it needs. When that cell is preparing to divide, each chromosome will be carefully copied so that each duplicated chromosome contains 2 sister chromatids, each with the same set of genes. But we actually have 2 copies of every chromosome, and therefore we have 2 copies of every gene. These pairs of chromosomes are called homologous chromosomes, but it is important not to confuse homologous chromosomes with sister chromatids. To understand the difference, we need to know something about these things called alleles. An allele is the specific form of a gene. For example, a chromosome may hold the gene that determines your blood type, but which specific blood type you have depends on your alleles. You could have type A alleles for the blood type gene, or type B alleles, or type O alleles. To use an analogy, if there was a gene for ice cream, you might have a chocolate allele, or a vanilla allele, or a strawberry allele. Here in this diagram, we see 2 pairs of homologous chromosomes, and because DNA replication has already occurred, each chromosome has 2 sister chromatids. You should notice that each sister chromatid on a chromosome has not only the same genes, but also the same alleles for those genes, as indicated by the color coding we see here. Pairs of homologous chromosomes also have the same genes, as indicated by these arrows, but they may or may not have the same alleles for those genes. For example, let's say that this top gene in this set of homologous chromosomes codes for eye color. On one chromosome, we find alleles for blue eyes, and on the other, we find alleles for brown eyes. Same gene, different alleles. Maybe the next gene down codes for blood type, and in this case we see that both chromosomes contain alleles for the same blood type. Over here, on these smaller homologous chromosomes, we see that there are different alleles for each of these three genes on the chromosomes. And over here on the bottom of our larger set of chromosomes, we can see another example of having the same alleles on both chromosomes. In both cases, each sister chromatid has identical alleles to the other sister chromatid. But these may or may not be different alleles than you would find on the second chromosome from that homologous pair, even if all the genes themselves are the same. Ultimately, the interactions between homologous chromosomes determines our traits, and whether we have brown eyes or blue eyes, when we may in fact have an allele for each. This figure depicts how these chromatids and chromosomes get sorted during cell division. At the top, we can see a pair of homologous chromosomes prior to replication. These homologous chromosomes have the same genes, but they may or may not have the same alleles. After DNA replication, each duplicated chromosome contains two sister chromatids. As the cell divides, these sister chromatids are separated from one another so that each new cell gets one sister chromatid, but also so that each new cell still has a set of homologous chromosomes. It is because of this sorting process that each new cell produced is genetically identical to the original cell. In humans, our 3.5 billion base pairs of DNA are organized into just 23 pairs of homologous chromosomes. This diagram is called a karyotype, and you should notice that the chromosomes depicted in this human karyotype are grouped into sets of homologous chromosomes, sets of 2, and that these sets are arranged mostly according to size, with the exception of this very last set, which is special because this last set determines our biological sex, whether we are biologically male or biologically female. This karyotype, which is a computer-generated, stylized version of the real thing, depicts unduplicated chromosomes, whereas this karyotype contains duplicated chromosomes, each with two sister chromatids. We can see that our study subject is biologically male because he has one X chromosome and one Y chromosome. All of this can be explained by the chromosome theory of inheritance, which helps us to better understand the way in which chromosomes, and therefore genes, are passed from parents to their offspring during sexual reproduction. During sexual reproduction, specialized reproductive cells, eggs and sperm, combine to form a new organism. Each human sperm cell contains 23 individual chromosomes and no homologous chromosomes. Human egg cells are the same way, containing 23 individual chromosomes and no homologous chromosomes. That means that egg cells and sperm cells contain only one of each chromosome, as opposed to having two of every chromosome like the rest of our cells. After the sperm cell fertilizes the egg cell, and a cell called a zygote is formed, these two half sets of chromosomes are combined to form one complete set, 23 pairs of homologous chromosomes, or 46 total chromosomes. We have two of every chromosome because we inherited one from our biological father and one from our biological mother. This is why our homologous pairs of chromosomes have the same genes, but not necessarily the same alleles. And this is also why we look kind of like our mothers and kind of like our fathers, but also look unique in our own way. We literally have both our parents' genes inside of our cells. These genes, which we inherit from our parents, instruct our cells how to build the protein molecules necessary to support life, and the function of these proteins ultimately determines our traits and our abilities. And because each new generation of organisms is made of a combination of genes and traits passed on from the previous generation, that means that each new generation of organisms is similar to, but also distinct from the previous generation. This is why doctors in particular care about things like family history, because genes that either contributed to or detracted from the health and well-being of your ancestors may have been passed on to you as well, and may therefore contribute to or detract from your health and well-being as a result. Understanding how genes, and therefore traits, are passed from one generation to the next helps us to better understand why certain traits run in families, and helps us to better understand why we are the way we are. And with that, I will bring our video to a close. Thanks for watching, and remember, you can go back and watch this video as many times as you need until you feel like you understand the structure and function of these important things called chromosomes.